we've got some wonderful guests here, just kicking off London Calling. You've been looking through the agenda, seeing what excites you. Yaw, I'd like to ask you first, what are you looking forward to most on the agenda today?
Well, it's my first London Calling, so excited to see the breadth of scientists and the breadth of people using ONT platforms across the world. As somebody based in Ghana in Africa, I'm particularly interested in networking with people using these technologies in emerging markets, in resource-limited settings. I think there's a lot of cross-pollination and cross-talk that can happen about how people are solving interesting problems in resource-limited settings using ONT technology. So I'm really excited about the opportunity to interact with other people who are approaching problems maybe in ways that would be applicable to what my company does in Ghana.
Fantastic. And Ivo, you were here on the sofa a year ago. I mean, what's changed? What are you looking forward to seeing on the agenda today?
Well, so first of all, I don't know how many London callings I've been to, but I've been to very many. And obviously each year, ONT puts something on top of what we had the year before. The thing that's always interesting, and this is the curiosity I have, is how ONT is going to essentially replace the presentation that Clive Brown used to give. This is truly going to be very different in terms of how it is sort of rolled out. The other thing is the same thing, networking with other people is crucial. And here you tend to meet everyone who is working on similar problems, and you sort of see how people are moving forward. For myself, we use nanopore sequencing in a very broad context, many different applications, and we try to push the newest applications that we can think of and try to put them on. And here it's always interesting to get someone, explain your experiment or what you're trying to do in one of your experiments to the people, and then you sort of get this feedback of, oh, have you not thought about? And you go, well, we did, but we didn't think that was reachable. And then someone says, no, but if you do this, then you can do it. So this interaction with our colleagues is definitely very important. Plus seeing, obviously, the things that are going to go onto the stage here.
Fantastic. Well, look, you're in an innovation and research environment in Ghana. Obviously, Ivo, you're in Barcelona, but you're on a plane all the time, going all around the world. Prasad's come all the way from Singapore. You're working in these amazing national genomics settings. What are you looking forward to on the agenda?
Yeah, this is my first experience at London Calling, and it's really good. And as an early researcher, this is a gold mine for me to get networked with the different people, to learn from how they are doing research. It's really good opportunity for me to learn from most experienced researchers and see how Nanopore is pushing boundaries in the genomic domains. Yeah.
Fantastic.
I'm really excited to see different speakers, talks, posters. Yes, I'm really excited to look forward to this event. Yeah.
So what is it you think that a national genomics program can perhaps learn from a cancer and innovation program in Ghana? How do you think that connection could work?
Yes, yeah. So in the genomic field, I would believe that sharing plays a vital role, and because such large projects require you to learn from many mistakes, what someone has done, we can learn from them to improve our projects. Yeah, so I would say yes. Not just the Ghana, or for example, Europe being a leading expertise in the genomics, we have learned a lot from them on how to proceed with such a large-scale human population genomics. Yeah.
Fantastic. Yeah. So all of you are working in sort of diverse data sets. Could you tell me a little bit about the importance to you of bringing diverse representation into the field of genomics? I mean, Ivo, you're working across a lot of European nations. How are you thinking about that?
Yeah, well, I mean, diverse, we have many applications, and this is a little bit the tricky thing, that you're handling 10 different ways of using nanopores, but we try to exploit it that way. Diversity in general. In Europe, we have one project that we're running at the moment. It's called the Genome of Europe, and we're trying to establish the genetic background of Europe so that we can use that to sort of build the personalized medicine system on top of this knowledge of the genome that is more accurate than just going through aligning against a sort of a very generic reference and then working. So you can get more precision by having better definitions, further reach out into areas of the genome, say human genome, that are very, very difficult to work with, repetitive regions, ends of chromosomes, centromeres, things like that, where obviously ONT has an edge technologically that you can reach into, that you can't reach into with the short read methods.
Interesting. And a lot of the work that you're doing is within the Africa Genome Atlas. Can you tell us a little bit about the work there and the importance of representation in global data sets?
Yeah, so I mean, with when we think about technology, sometimes we can get so excited about it. We may be tempted to lose sight of the fact that at the end of the day, what we're trying to do is improve people's lives, improve the health of people. And unfortunately, historically, genomic data sets have been skewed with certain populations underrepresented significantly. People of African descent are one of the most underrepresented, with less than 3% of genomic data representing them, whereas people of European ancestry have about 80% of data. So what we're trying to do at Yamachi is address that gap by building what we call the African Cancer Atlas, which is focused on a Pan-African clinical genomic database of people with cancer across Africa, to really try to better understand the genetic basis of cancer in these populations. Because, as I said, the goal is, we want to improve people's lives, but you can't do that if your data represents a very small slice of humanity. And so what we're trying to do is, how can we make genomics work for everyone? And it starts off by making sure that you have enough data from enough of a broad spectrum of people that your applications are useful in those populations. And what I like about ONT is, ONT is built around accessibility, democratizing sequencing. But if you just simply democratize in London or in New York, then you're missing the point. You need to be sequencing in Accra, in Lagos, in Singapore, in New Delhi. And so that's what excites me about this community, is you have people trying to use this technology in their locales to sequence people around them. And then ultimately, the world gets a very, very diverse database. So we're trying to do something for Africa, and I think it would mirror what is being done in Europe, what is being done in Asia. And then together as a community, we have a much better picture of human genomes, not geographically specific genomes, which will then advance medicine everywhere.
I have a follow-up question, but I also need to remind the audience, you can ask questions on Slido. You can basically scan the QR code and send in a question for the audience I've got here today. So just before we move on, you mentioned capital cities just then. Are you seeking to take cancer diagnostics out of the capital city and into even the rural regions?
I think that would be the ultimate goal. Right now it's all about whether you're in big, well-developed countries or in resource-limited settings, there's a lot of centralization. So rural areas of the United States have issues with cancer diagnostics. Of course, in Africa, it's well documented, the challenge we have with access. So I think that would be the dream. But to be honest, you can't do that unless you have a good understanding of the basic genomics of the population. So, it's a stepwise process. You can rush to try to take a diagnostic to a rural area, but how do you know that diagnostic will actually work in that population if you haven't developed it with data from people who have similar backgrounds? So that is the ultimate goal, and look forward to seeing that happen as we continue to progress.
Well, that will be a common theme over the next 2 days. A lot of people here talking about the foundation of discovery, the process of method development, the translational process, and then ultimately implementation. So in Singapore, you're not just working for the Singaporean population there. The Precise program has 3 different ethnic groups included. Can you talk a little bit about that?
Yes. So the current, what we are focusing on is building a pan-genome references to capture the diversity between these 3 population. So as you can see, most of the large-scale projects, what we have is European-based. And South Asia or Asia represent at least 40% of the world's population, and it is very well underrepresented. So one of our focus is to close the gap of that underrepresented population in the genomic domain. So our focus is local relevant, and yet how can we contribute to the global genomic domain? So that's what our focus is. Yes.
And the Singaporean system has got a really well-structured process for moving from discovery through translational. Is that correct? Can you tell us a little bit about that?
Yes. Yeah.
Great. And Ivo, how is the Genomes of Europe program thinking about that translational journey and making connections within more innovative communities that develop those applications into diagnostics?
Yeah, so the same as my colleagues, we actually have the same problem. We all, it might be conceived that Europe has a very level playing field, but actually that it's not as level as it actually should be. So we have to level our playing field as well. And I think it's just it's sort of a process where you do this, but at the same time you show how it can be done, and you hopefully help other centers that are trying to level their playing field as well in terms of technologically, methodologically, how you actually do that. So I see it that way, that we're viewed as we have an advantage, but it's actually not quite as big as we would hope it would be.
Yes.
And I think this is sort of the objective of our project very much. I have to say one thing, and this is where I really like the African projects. I actually have one researcher of my center in Senegal doing rare disease research, and this is really quite amazing when you sort of start connecting with the technological things that you have with a country that has a totally different way of organizing its healthcare. And you sort of see the impact that you can have by bringing your technology to the other place. So this is something we probably should talk more about that.
Yeah, happy to do that.
Well, I can see that this conversation could go on for a lot more, and this is exactly why we bring people here into the studio. We've got Gordon's opening talk coming in a minute. We're so excited to see so many more speakers coming into the studio, and thank you to the guys here for sharing their perspectives. So fill up your coffee cup, get ready. The big talk is about to begin, and we hope you tune into a lot more sessions. Thanks for joining us today.